Welcome back, Mindsetters. I'm glad that you guys are on the ball and you guys are enjoying this lesson. I know it's awesome for all that stuff that you may have forgotten to be put back into your awareness so that on Monday, when you write, or any other day perhaps, you know what to do. Remember that there's also winter school booklets that are given away to the first 5 callers that have questions today, and good questions. Our number that's on Facebook as well is 086 105 8262. You can call this number during the entire show just so that we have your questions and Mr. Aslam can answer them right on the show just for you. Over to you, sir.
Thanks, Katlego. Welcome back, Mindsetters. Also from my side, please post your questions on the Facebook page as well as call in. If you have even a particular question in a particular year that you want us to go through that we don't have on the screen right now, we would then put that on the screen and answer it for you as well. So please phone in with your questions. Another tip of advice as well, people, remember when you are studying, make sure that your teachers have given you what is called an exam guideline. This is not a state secret. It's an official document which any learner can have access to. You can even find it probably on the departmental website. But if not, you can get it from your teachers. They have it with them. Ask them to forward it to you so that you know that you are learning the right stuff. Remember, at the end of the year, there are 2 versions of the paper. Version 1, which is the current grade 12 full-time candidates that write, and version 2 is for those learners who didn't make it in 2010 and they're rewriting again. So please make sure that you know which version you are writing, and for that you need the document called the exam guideline, so you know you're not messing up in terms of what you're learning. Just to move on to paper 2, because the requests are coming in fast and furious regarding paper 2. So we're moving to paper 2. And just to quickly recap what is in paper 2, the first one is plant hormones. Then we look at animal or human hormones, or the endocrine system. Then we move on to the nervous system. And here we talk of the central nervous system, which is the brain and spinal cord, and the sense organs. In the sense organ, we only focus on the eye. Mind my pun on focus. Focus on the eye and the ear. Those are the only 2 sense organs that you're going to need to know for this year. We move on from there to reproduction in humans, the topic you're probably all waiting for. Reproduction in humans and plants, and also some reproductive strategies in other animals. So that is where we're moving with that. We also finish there, the human part, with thermoregulation. That means the skin and what the skin does to keep us warm when it is cold, to keep us cool when it is warm. And thereafter we go to environment. And in environment, you learn about population environment or population studies and social environment issues. We're talking about how organisms have a social network and what do they do to look after each other in the environment or in the wild. So this is basically what covers your paper 2 items. So you need to know these things for paper 2. It's longer in some ways than paper 1. So let's go to our first question here. Now we've put this particular question more for the type of question than the content, because this covers all the content itself. But the important thing is to understand this question. The question is very clear. It says indicate whether each of the statements in column 1, that's column 1, applies to A only, B only, both A and B, or none in column 2. So all you're going to do is you're going to write on your answer book 1.3.1 and next to it the answer. And your answer can either be any one of these bold characters here. A only, B only, both A and B, or both A and B, or none of the items. Please do not rewrite these statements in your answer book. You would not be credited marks for that. Let's go straight into the question. A tube that stores sperm cells until maturation. Now here, in an exam situation, we would have to specify probably in humans. Here's an example, so we did not specify that. But in an exam situation, we would have to say the human sperm, in that case here. And in that case here, our answer is it stored in a vas deferens? What's the vas deferens again? The sperm duct. What's the job of the sperm duct? What are we testing here? We're testing whether you know the functions of the different parts in the male sex organs. And if you don't know that, you're in big problems. Now, vas deferens transports sperms from the testes through all the storage there, right up past the seminal vesicles, prostate gland, and so on, and eventually into the urethra where it's going to be ejaculated. So, vas deferens does not store sperm, so it can't be A. The seminiferous tubules, guys, where are the seminiferous tubules? These are the tubules that make up the testes. These are the tubules that increase the surface area for the production of sperm. So in this particular case, obviously, your answer is none. Okay? The next one, the cell division that takes place in the gametophyte generation to form gametes. In the gametophyte generation, to form gametes. Very important clue here, to form gametes. Which one of these two is involved in the formation of gametes? By now you should know that, folks, and that is obviously A only. Mitosis is not there to form the gametes. Remember, however, in the formation of gametes, there may be some mitosis taking place, but to form these gametes, meiosis is chiefly responsible. Now, if we have this type of a question, at the end of the year, we may run into lengthy discussions, memo discussions to finalize the answer in that particular case. Obviously, before that, we probably avoid that type of a question, because the learner may well say that there is a bit of mitosis even in the formation of the embryo, etc. But here they specifically saying to form gametes. All right? Controls all voluntary action. Controls all voluntary action. And here your answer is A, cerebrum. Controls. Now remember, the cerebellum doesn't control it, it coordinates it. The fact that I can lift my hand up and down and throw it around, whatever, that is controlled by the cerebrum. But the fact that my hand is not just going up and down without any coordination, the fact that if I throw this pen up, I'm not going to do that now because it falls and I'm in problems. If I had to throw this pen up and catch it, okay, I'll do it anyway, okay. The fact that I could throw the pen up, cerebrum. The fact that I could follow it up with my eye, and my eye and hand coordinate to catch it again, cerebellum. Coordination of the voluntary actions. Okay, the last one there, body temperature varies with the environmental temperature. That means as the environmental temperature changes, the body temperature also changes. In this case here, it will obviously be B only, ectotherms. Endotherms, they are those that generate their own temperature from inside their body. Ectotherm, as it says, ecto from outside. They depending on the environment. Therefore, you will see most ectotherms in winter, etc., they become less active because the body temperature drops, and in terms of saving or conserving energy, they drop their metabolic activities to a bare minimum just to survive. Good, you'll notice there are 2 marks for each question here, and you can really score. Last year, end of the year 2011, and the March 2012 paper had many more of this, plus minus 7 or 8. So that's 16 marks, guys. 16 whole marks. You cannot mess up this question, and it's fairly easy. You need to know your work, though. Let's move on and straight into plant hormones. Now here again, I love these type of questions where they deal with investigations. A group of grade 12 learners carried out an investigation to determine the effect of gibberellin on the germination of the seeds. Remember, for your work, you need to only know the functions and functioning of auxins, gibberellins, and cytokinins. Those are the only 3 that you need to know. There are others in the textbook. You only have to worry about those 3. Okay? The following procedure was followed. This, by the way, comes from March 2012. A sample of hazelnut seeds were divided into 2 groups A and B. A gibberellin solution was added to the seeds in group A. Water was added to the seeds in group B. Both groups of seeds were allowed to germinate for 16 days. The percentage of seeds germinating in the 2 groups was recorded, and here are the results in a nice little graph. Okay? On the y-axis, the percentage germination, how well did they germinate. On the x-axis, we have time, independent variable, dependent variable. I will keep talking about independent and dependent because you need to know it when you're doing hypothesis, when you need to know it when you're doing investigations, you need to know it when you're drawing a graph. The independent variable goes on the x-axis, the dependent variable on the y-axis. So keep remembering that all the time. And they give you a key. Notice also when you're drawing a graph, you make sure that you have a key as well. They're showing you here the one with the diamond is gibberellin, the one with the green squares, or squares I think, is the one that was put in water. Are you with me? The first question says, formulate a possible hypothesis for this investigation. Let's go back to the investigation itself. What did they do? They wanted to know the effect of gibberellin on the germination of seeds. So obviously the hypothesis must hinge around this. But also note that they kept seeds in 2 different conditions. And what are the 2 characteristics? Or rather the variables. The variables are the percentage germination. The percentage germination, let's go back to that graph. Here's the graph. The percentage germination, one variable. The other variable is time in this graph. But it wasn't time that we were checking. We weren't leaving them there on their own and checking against time. What did time do? We were worried about one in gibberellin and the other one in water. So our hypothesis must have these things in them. So we can say that the percentage germination, that will give you the 1 mark, of the seeds, etc., increases, or you can write decreases, when placed in gibberellin or water. Remember, this is a hypothesis. It's not based on the result. You formulate a hypothesis before you investigate. So can you see we got slashes here? So if the child wrote increases or decreases, you're still going to get a mark. If he wrote gibberellin or water there, you're still going to get a mark. If you put a question mark at the end of this, you get 0, even though you got all 3 variables, because a hypothesis is not a question. So please remember that. All right, so that was the first one. Second one, using the information from the graph, determine the percentage germination of hazelnut seeds on the 10th day. Let's go back to the graph. Simple question. Go to the 10th day. Look at this thing here, 4, 8, 12, 16. So 8, 12, there's a gap there, that should be 10. So you go up this line, up to there, somewhere, and obviously you're somewhere in this area here. Notice you are not on the line where it says 80, so your answer cannot be 80. It's not halfway between 70 and 80, so your answer cannot be 75. Your answer has to be above 75, and it's closer to the line, so it should be between 77 or 78. That is your answer, and it's percentage, because sometimes they give you 2 marks, you can do that. 1 mark in this. Explain the purpose of Group B. Group B were put in water. What did we want to know? Whether gibberellins affects the growth of plants. And we kept 1 in water. What did we do? We excluded gibberellins. So what does that become? That becomes the control. Why do we have a control? To compare our results, and we want to know that it's gibberellin indeed that caused the change and not anything else. So if we put it in water, that's what we can understand there.
Quick break. Let's do that. I know that there's a lot for them to take in and to, let it simmer in your mind a little bit while we take a very, very quick break.